Hello, my name is Tim and I'm a Business Development Manager with Synbio Technologies. I'm here today to talk about custom DNA oligos. We will cover how they're typically synthesized, purified, and some exciting ways to use them. Let's start with a general workflow for oligo synthesis. Step 1 is to confirm the desired sequence of the oligo. Step 2 is to perform the chemical synthesis in order to manufacture the oligo. Then we'll cleave the oligo from the solid support and remove the protecting groups. We'll purify the oligo using the desired method, perform the quality control steps, and finally deliver the final product to you. Oligonucleotide synthesis follows a cycle. The first step depicted here is called detritylation. The DMT protecting group, highlighted here in red, is removed with an acid solution such as 2% trichloroacetic acid or 3% dichloroacetic acid in an inert solvent. This results in a free hydroxyl group. The next step is coupling. This is where the amidite is activated with a mild acidic solution. Next, the amidite is added to the existing 5' hydroxyl group of the previous base. Since this chemical reaction tends to be around 98% efficient, the non-reacted groups must be acetylated with a capping solution, such as acetic anhydride or N-methylimidazole. This prevents additional amidites from being added in later reactions, thus preventing the formation of mutated oligos that contain deletions, as these would be nearly impossible to purify. After capping the unreacted oligos, the product is an unstable intermediate with a phosphate triester. With the addition of iodine, the phosphate undergoes an oxidation reaction, generating the stable tetracoordinated phosphate triester. At this stage, the DMT cap can be removed, and the cycle can repeat to continue growing the oligo. When oligosynthesis is completed, the attachments to the support are released with a strong base, and the cyanoprotecting group can be removed at this stage as well. However, the protected oligo can also be released and used for cartridge purification, which we will demonstrate on a later slide. Now that we know that the coupling reaction is not perfect, this chart helps illustrate the theoretical yield for complete oligos depending on the length of the oligo and the reaction efficiency. The industrial standard is 98.5% efficient, where at Synbio our average efficiency is 99.3%. This means less truncated oligos in the final product and higher synthesis yields per reaction. Here are the four most common options for purifying synthesized oligos: desalting, reverse phase chromatography or cartridge, high-performance liquid chromatography, and polyacrylamide gel electrophoresis. Desalting is generally the default purification method for oligosynthesis. Desalting is suitable for PCR primers and sequencing primers. The product of the oligosynthesis is loaded onto a solid substrate. Using size exclusion, the byproducts of the reaction are trapped within the pores of the column, while the oligos can be rapidly eluted. This method effectively removes salts and other contaminants, but truncated oligos will still be present in the final product. Cartridge works by taking advantage of the hydrophobic nature of the DMT cap. The completed oligos will have the DMT cap still present from the last addition of the amidite, while all the other oligos will be acetylated at the 5' end. When added to a solid support material such as C18, the acetylated or incomplete oligos will wash away readily. Then, the completed oligos can be eluted and deprotected for final use. For HPLC, the oligos are run through a column under high pressure. This method is suitable for qPCR probes, antisense oligos, and in vivo applications where purity is of great concern. The two standard columns are a reverse phase column and an anion exchange column. The anion exchange column takes advantage of the negatively charged phosphate backbone, so the electrostatic interaction between the positive charge of the column and the negative charge of the oligo slows down oligos with a greater charge. This is effective for separating oligos in the 10 to 40 base pair range. For reverse phase HPLC, the column is using the hydrophobic nature of larger oligos to separate out the unreacted oligos. This is more effective for oligos in the 10 to 50 base pair range, especially those with modifications, as these tend to make the oligos much more hydrophobic. Here is an example chromatogram from an HPLC run. Highlighted in blue contains the final product, which can be recovered at that time. Page gel can resolve oligos at a resolution of 1 base pair. This method is suitable for oligos larger than 60 base pairs that still require high purity. And this method can be combined with HPLC to provide the highest levels of purity. However, this method can be incompatible with some modifications due to the acids used during running the gel. The oligos are loaded onto the polyacrylamide, and a current is used to pull the oligos through the gel. The larger fragments take longer to get through the gel due to more time required to make it through the pores of the gel. Finally, the correct band is cut out of the gel and purified to provide the high-quality oligo at the specified base pair. Now that we know how oligos are made and purified, let's look at some of the more common uses for oligos. Antisense oligos, or ASOs, are a relatively new tool for researchers to knock down protein expression. They are small oligos, usually around 18 to 30 nucleotides in length. Their sequence is a reverse complement or antisense to the target RNA strand. So, in the cytoplasm of the cell, when this ASO encounters its complementary RNA, they'll bind together. Depending on the backbone of the ASO, RNase H can recognize the DNA bound to the RNA and digest it. Either way, the RNA is unable to be translated, thus reducing the corresponding protein expression. Some common modifications for ASOs include locked nucleic acids, modified bases, and 5' end modifications. Locked nucleic acids have an ester bound that locks the ribose in place. This makes the DNA more resistant to enzymatic degradation and may improve the affinity of the ASO. One example of modified bases is phosphorylthioates. These are DNA molecules with the oxygen here circled in red replaced with the thiol group. This again helps prevent enzymatic degradation. Finally, 5' end modifications such as using GalNAc as depicted here can increase the amount of ASO uptaken by the cell. Now for one of the most common uses for oligos, primers. Primers are used in PCR to enable polymerase to bind to specific sites on the DNA. One cool thing you can do with primers is using overhang primers. Here is the DNA. Now we can add the primer, and it has a region where it is the reverse complement to the DNA. This we'll call the binding region. The part hanging off is called the overhang region. Note it is depicted here that it is at the end of the DNA strand, but the binding region can be in the center of the DNA strand, and this will still work. The polymerase then binds and synthesizes the antisense strand, and the cycle can repeat itself with a reverse primer and then completes the DNA synthesis. You can now see that the DNA strand is longer and incorporates the overhang region. Probes go hand in hand with primers for quantitative PCR. Probes have a fluorescent molecule that is covalently bound, usually at the end of a short oligo. When the polymerase tries to add nucleotides through the probe, it has exonuclease activity, meaning it will release the probe's nucleotides individually into the solution. So, the nucleotide with the fluorescent molecule is then free from its tightly bound vicinity with the quencher molecule as depicted here in black. Now, when the probe is energized with a laser, it can emit light that a detector can read. For gene fragment synthesis, I will link a more detailed video in the description below. The basic concept is to create a multitude of oligos that span the entire gene fragment. These oligos have complementary regions on the ends of each other, so they can bind to one another. The gap then acts as priming sites for polymerase binding. After the polymerase reaction is completed, the resulting DNA is your full gene fragment. Some of the key advantages of working with Synbio for your oligo needs are, we pride ourselves on quality. We have both ISO 9001 and 13485 qualifications, which means our documentation and quality control procedures are reviewed by a third party. We can deliver specific amounts requested or deliver by synthesis scale, and we offer a full range of purification methods. We have specialized scientists for each one of our product lines, so your technical questions will be addressed by an expert in the field. And our mission is to empower scientific discovery and innovation through our advanced one-stop DNA manufacturing and design solutions with an emphasis on affordable prices. At Synbio, we operate over 20 Dr. Oligo synthesizers of the 192 model and 4 768 models, meaning we can synthesize over 6,000 different oligos at a given time. This is a non-comprehensive list of the modifications we offer. Some highlights include locked nucleic acid, multiple fluorescent dyes, and modified ends for click chemistry. When you submit an oligo to Synbio, it will be classified under one of our 4 major services. Our standard service, our long oligo service where we can synthesize oligos up to 150 base pairs, our microscale oligos for small batches, and our large scale oligos where we can synthesize up to 1 gram of the desired oligo. To generate a quote for your oligo synthesis project, go to our website and create an account. Here you'll find a quick order list where you can click on Oligo Synthesis, which will bring up this form. In this form, you can fill out the required info for your oligo synthesis project, and once complete, click on Add to Cart. This will bring you to a verification page where you can ensure the sequence and the other information is correct. Once you click on Submit, a project manager will get back to you within 24 hours. Once your oligo is prepared, we'll perform mass analysis to ensure the oligo is of the correct size, and this report will be included in your shipment. I hope you learned a little something about oligo synthesis. My name is Tim, and if you have any other questions, feel free to reach out to me at timothy.acri@symbio-tech.com or leave a comment down below. Thank you for your time, and I hope you have a wonderful rest of your day.